Hello and welcome to our channel A Showtime. In this video I am going to discuss a very important part of biochemistry, which is protein sequence. You will definitely get 2 or more questions from this topic in CSIR NET exam. After this tutorial you can easily solve them. I compiled few questions which are asked previously in CSIR NET exam, and I am going to tell you how to use provided information to solve the questions quickly and get the right answer. First of all, you need to know about amino acid. There are 20 essential amino acids, and in the primary structure of protein, amino acid residue attached via peptide bond. So you need to learn the single letter annotation for these amino acids, which is quite simple, and a table is provided in this video. After that you need to know the primary structure of protein. So these are the single letter annotation for 20 amino acids given in this table. Going on our next slide. This is the primary structure of protein. This is the N terminal or amino terminal, and this is the C terminal or carboxy terminal. Now you should know how the cleavage process takes place. Basically it is of two types. One is terminal cleavage and the other is internal cleavage. The terminal cleavage which occurs on terminals either on N or C terminal, and other is internal cleavage which do not occur on the terminals but anywhere in the chain. So the internal cleavage can take place either there, there or there, but not there and there, because these sites are secured for terminal cleavage. There are certain reagents which cleaves the protein. Some of them perform terminal cleavage and a few perform internal cleavage. Another very important table is provided in this video, which starts from there. These are the reagents and they are categorized based on their nature. So going on, there are few which perform terminal cleavage. Adamant degradation, which cleaves C side of N terminus. That means this is the N terminus and the C side. That means right hand side. Which is the right hand side? This is the right hand side of this N terminus. So adamant degradation cleaves the C side of N terminal. This is the N terminal and this is the C side of N terminal. When we use adamant reagent, it will cleave this particular bond, and the other are carboxypeptidase A, which cleaves N side of C terminus. That means the carboxy terminus and the left hand side. And R should not be arginine, lysine or proline, and the Rn-1 position there should not be proline. Otherwise there will be no cleavage by carboxypeptidase A. So as in carboxypeptidase B, it cleaves N side of C terminus. R should be either arginine, lysine or amino ethyl cysteine, and on the Rn- position there should be no proline. With carboxypeptidase C, Rn is equal to any amino acid. It will cleave any amino acid on the terminal, but this should not be proline. And there are others, and the details is given in this table. You can take notes from this table or you can do a screenshot. these reagents, they perform internal cleavage. CN bromide, which is cyanogen bromide, it cleaves C side of Rn. That means I can describe you. Suppose this is the R, that is methionine, cleaved by cyanogen bromide. So cyanogen bromide will cleave this side, which is C side of Rn, not this side. C side means right hand side bond. So when we apply cyanogen bromide, it will cleave this specific bond, which is in the C side of methionine. So as trypsin, it will cleave C side of Rn, and Rn can be arginine, lysine, but Rn plus 1 position, there should be no proline. If the proline is present in Rn plus 1 position, there will be no cleavage. In chymotrypsin, it also cleaves C side of Rn. Rn can be phenylalanine, tryptophan, tyrosine or leucine. But on Rn plus 1 position, there should be no proline. And you can understand the other reagents as well in this table. So here comes few questions you can solve with the help of given information. I am going to solve these questions, and you can see how you could use the given information to solve the question in your exam. So the question is, which one of the following proteases does not cleave on the carboxyl side of any arginine residue protein? So you need to know that it is given in table that trypsin, proteinase K, thrombin and chymotrypsin are the options. So the correct answer is D, which is chymotrypsin, and I am going to explain how. The correct answer is chymotrypsin because it cleaves C side of phenylalanine, tryptophan, tyrosine and leucine, but not of arginine. Apart from this, trypsin cleaves for arginine, and proteinase K and thrombin also cleaves for arginine, but not chymotrypsin. So the correct answer is D. Going on our next question, which is the peptide alanine, arginine, glycine, methionine, threonine, tryptophan, lysine and valine was digested with cyanogen bromide to produce which one of these sequence? So as you know, it is given in that table that cyanogen bromide cleaves the C side of methionine. So the C side of methionine is this. Cyanogen bromide will cleave this side. So you will get two sequences. One is alanine, arginine, glycine and methionine, and the other will threonine, tryptophan, lysine and valine, and you can see the correct option in there. The correct option is answer A. So the correct answer is A, as you will get the sequences like alanine, arginine, glycine and methionine. This bond is cleaved, this bond is cleaved, and the remaining is threonine, tryptophan, lysine and valine. So you can solve this question very easily, and these questions are asked in CSIR NET exam. Next question. The oligopeptide, which is composed of given amino acid phenylalanine, alanine, arginine, proline, methionine and this is tryptophan, serine, arginine, proline, glycine and phenylalanine is treated with trypsin, chymotrypsin and carboxypeptide is B. So apart from the original peptide, the number of fragments obtained will be. So all you need to do is perform the cleavage by these reagents. So first is trypsin. It is given in that table that trypsin will cleave the C side of R, and R can be either arginine or lysine. So you can see this is arginine. So trypsin will cleave C side of arginine, but the condition is that there should be no proline at n+1 condition. You can see there is a proline at n+1 condition. So trypsin will not cleave this side. So there will be no cleavage performed by trypsin. Next is chymotrypsin. In case of chymotrypsin, it cleaves phenylalanine, C side of phenylalanine, and it should be somewhere in the chain, not at the terminal. You can see this is terminal phenylalanine, this is terminal phenylalanine. So chymotrypsin will not cleave phenylalanine as it is situated at terminals. Going on next, which is carboxypeptidase B. Carboxypeptidase B also cleaves it cleaves arginine, right? Carboxypeptidase B cleaves arginine, but there should be no proline at n-1 position. So you can see, and carboxypeptidase B also performs terminal cleavage, so it will not cleave this chain. So the answer is 0. As you can see, trypsin will not cleave it because there is proline present near arginine. Chymotrypsin will not cleave it because it cleaves internal cleavage, not the terminals, and carboxypeptidase B will not cleave it as it performs the internal cleavage as well, because there is no arginine at the terminals. So carboxypeptidase B will not cleave this chain, so you will get no fragments at all. So the answer is D, which is 0. Next question. As you can see, the language is pretty heavy of this question, but the solution is really easy if you can read the question, which is: in order to determine the primary structure of an octapeptide, amino acid composition was determined by acid hydrolysis A. The intact oligopeptides were treated with carboxypeptidase, and you will get this B, this structure. When treated with chymotrypsin, you will get this C composition. Treated with trypsin, you will get this composition, which is B, and when treated with C and Br, you will get this composition, which is E. So all you need to see is that on treatment of these given reagents and the composition, if it fits their nature of cleavage. So at first, there is the first one, which is carboxypeptidase B and carboxypeptidase, and it gives this chain, which is B. As it is not specified that either it is carboxypeptidase A or B, you cannot predict the answer, so you will ignore the carboxypeptidase right there. The next one is chymotrypsin, and it gives the composition C. So as you know, the nature of cleavage of chymotrypsin is C side of Rn, and Rn could be either phenylalanine, tryptophan, tyrosine and leucine. So in C chain, you can see this is the cleavage portion, and there is no given amino acid which are cleaved by chymotrypsin. There is no phenylalanine, no trypsin, no tyrosine, no leucine. So you will reject this C. Next is trypsin, which gives this D composition. So trypsin, as you know, trypsin cleaves C side of arginine and lysine. So you know the cleavage point is this, near the arginine and the C side of arginine. So this fits, and this cleavage point can be near the lysine, as you can revert this sequence. Serine could be there, then phenylene, then lysine. So this is also between lysine. The C side of lysine is cleaved. So the correct sequence is D. It fits the cleavage by trypsin. So the correct sequence is D. As you know, this arginine, this is the C side. You know, this is the answer. Alanine, arginine, then serine, phenylalanine, lysine, then methionine is there, serine is there, and then alanine, which is there. So the correct order of this octapeptide is alanine, arginine, serine, phenylalanine, lysine, methionine, serine, and alanine. And if we does the cleavage by trypsin, trypsin will cleave this side and this side, and you will get this sequence, and the correct sequence is this one. So you can easily solve this question by the given intel and easily get the 4 marks, and it is no big deal for you. So here are a few questions you can try to solve by yourself and give the correct answer which you think is correct in comment box, and I will provide the correct answer with solution very soon. These are the 2 questions. You can try to solve it on your own. Thanks for watching this video. Please like and share and subscribe our channel to get more and more information regarding CSIR NET exam of life sciences. Thank you so much.